Okay, well, I think we can get started, and a few people will trickle in as I do the several minutes intro. So, welcome everyone. Happy Friday, and thanks so much for coming to our monthly research seminar. We have a very exciting speaker today who I will introduce in a bit. But first, I would like to just remind us all about the Artificial Intelligence Precision Health Institute Affinity Group at the University of Hawaii, where our goals are to hear from leaders in the field of AI for healthcare, in particular for cancer informatics. We like to bring together an interdisciplinary group of researchers spanning biomedical informatics, computer science, nutrition, epidemiology, radiology, genomics, in terms of research, but together with clinicians and advocates as well, in order to foster collaborative interactions. So the format of these talks is a roughly 30 to 40-minute presentation, but it's loose. It can go shorter or longer, and then an interactive Q&A session afterwards. We have an exciting lineup of speakers, and I apologize, this slide is slightly out of date, but we are always welcome to feedback or suggestions for speakers. So if you have that, just feel free to send it to Dr. John Shepard or myself at the email addresses listed here. This seminar series is hosted by a few institutions within the University of Hawaii. So one of those is our Cancer Center, located in the Kaka'aka location, beautiful campus shown here. The Shepard Research Lab at the Cancer Center, as well as the Hawaii Data Science Institute that houses a lot of the data science work within the UH system broadly. We always like to have a paper of the month in the seminar series, and I think this might be one that our speaker today might mention. It's a really interesting paper that describes the first AI ML solution that assesses individual risks related to immune checkpoint inhibitors, but using structured EHR data that's collected just through routine practice. So it doesn't require additional data collection or documentation in the clinic. So interesting paper, definitely check it out after this seminar. So Dr. Osterman, we always like to give a speaker compensation for your time today. So after this talk, we'll be contacting you for your address, and we'll ship you a beer stein branded with AI Precision Health Institute logo from the University of Hawaii. And we love pictures of you with the beer stein. Doesn't have to be beer in the stein, but with the mug. So without further ado, I'd like to introduce our speaker today. So Dr. Osterman is a practicing medical oncologist, informatician, as well as the Associate Vice President for Research Informatics at the Vanderbilt University Medical Center, where he holds many hats, including as Director of Cancer Clinical Informatics. He has dual faculty appointments at the medical center in both the Department of BMI, so biomedical informatics, as well as the Department of Medicine in the Division of Hematology and Oncology. Dr. Osterman's clinical interest within oncology is for lung cancer, and his research focuses on applying clinical informatics methods across the cancer care continuum. So without further ado, I will stop sharing my screen, and Dr. Osterman, take it away.
Thank you, Peter, for that incredibly kind introduction. Let me share my screen here, and I will say that I am looking forward to the beer stein, but I may end up in more of the tequila category. But this is fun. I'm really excited to come and share with this group. I'm going to talk about predictive AI models and really data standards in action, because I think at the core of what we do is the data, and the better the data are, the better models and predictions ultimately we can make on top of that. And so I've been incredibly lucky in my institution to work in groups that have put a lot of effort into structuring data. So I want to highlight some of those efforts, and then we'll culminate with the paper that Peter highlighted. But I want to start by turning back the clock a little bit and just share a little bit about my origin story. So over 20 years ago, I was an undergraduate, and Frank Witzman was the PI in a laboratory at Indiana University, and I was had no idea that informatics was even a field, but was just probably more out of luck than anything, studying both biology and computer science in undergrad. And one of my advisors said, "Hey, there's a guy downtown doing proteomics, and it's in this field called informatics, and I think that you'd probably get along. You should connect with him and see if he has a spot in his lab." And I thought, "Oh, this is great. I'm going to be doing all this cutting edge computer science and biology." And what I signed up for ultimately, I guess, was to be on this right side of the diagram, was to pipette. And if any of you are from the basic science side, and I understand now because you can only trust an undergraduate to move their thumb about this far. So that was my job, was to pipette basically all of these little dots that we would cut out and digest from these 18 centimeter agarose gels into small little holes in this plate. And it was the most boring thing I could imagine. But I looked into the corner and I talked to one of my postdocs and I said, what's this thing in the corner? And he said, "Oh, that's the robot." And I said, "What does the robot do?" And he said, "Oh, the robot pipettes and does other things." And I said, "Oh, that's interesting because I pipette." And he said, "Well, the robot actually was paid for on a previous grant. And when the grant ran out, we couldn't pay for our service contract anymore. So the company doesn't come out and reprogram the robot. And so it's just been sitting there for the last year." I said, "So the robot works, you just haven't been able to reprogram it." And so I thought, "Oh, that's interesting." And after lab hours, I went online and grabbed the manual for the robot. And sure enough, there was a serial cable back tucked in away behind the robot. And I was able to hook to one of the computers and was able to program that robot to basically replace me as a summer worker and improve the lab efficiency. And I say that because I think it's really been a theme for my career. I don't know that I've ever had a novel idea in my lifetime, but I'm pretty good about looking around and finding the dusty robot in the corner and saying, "Hey, hold on, can't we make that work? And wouldn't that make everyone more efficient?" And that's really the theme of, I think, this talk and largely what I've been focusing on. The next summer, I went back to a genomics lab that was actually just down the hall, and that ultimately got bought by through a tech transfer from the Indiana University School of Medicine to a small company that ultimately called themselves LabRat Solutions. We were a laboratory information management system to help the workflow of laboratories. And so I've also always been interested in kind of this intersection between health IT, academics, medicine, and business. This was not one of those famous Silicon Valley startups. This was one of those that petered along for about 12 years and then ultimately folded. But it was a lot of fun. I had the opportunity to be employee one and work in that setting. So did that right before going to medical school, got my master's in biomedical informatics as I was a fellow here at Vanderbilt, and then stayed on as Vanderbilt as faculty. Several disclosures, pertinent ones for this talk. I'm going to talk about our collaboration with GE Healthcare, who funds some of my research. And so that's all very transparent. Everything else here I don't think necessarily applies, but they are here. I'm still trying to figure out what I do, what I want to do when I grow up. And so, like I said, I'm interested in kind of education and health IT research, administration. I do some things nationally, and I still have clinic one day a week where I see lung cancer patients. And I do so at Vanderbilt University Medical Center, which is located in just outside of downtown Nashville. We have about 40,000 employees and about 200 clinics. We encompass 3 different community-based hospitals in addition to our main campus university hospital. For the cancer center, the Vanderbilt-Ingram Cancer Center, the VIC, we see about 7,000 new cancer patients each year, totaling, I think we're up to now 200,000 outpatient visits per year. So this is where I practice. And again, I feel incredibly lucky. And today I'm going to structure my talk in these kind of 3 core areas. And as Peter said, I'm going to aim for about 40 minutes, and then hopefully we'll have a good discussion at the end. Since you all have a cancer center and your cancer center is co-sponsoring this, I always just like to highlight our data science shared resource because I think it's incredibly enabling in our cancer center, and I like to share how we're kind of structured. I want to talk a little bit about mCODE. I chair the mCODE Executive Committee. mCODE is the minimal common oncology data elements. And again, I think data is the foundation for all we do in AI. And then I'll end with the predicting and toxicity and efficacy efforts for immunotherapy. So we'll start with the CCSG data science core. So we're an NCI designated comprehensive cancer center, and our data science shared resource is a core within that cancer center. We are an incredibly large team. This photo doesn't include all of us, and if you look closely, you can see we've attempted to Photoshop some people in here as we've added folks recently, but it still doesn't encompass everyone. But it's both informatics and biostatistics. So I'm in the middle. I run the informatics side of our data science shared resource, and Yushir to my right in the picture runs the biostatistics portion. We largely serve a couple of roles. So I help develop the IT strategy for the Cancer Center, and then also serve as the informatics lead on most of our larger projects. So think U level and higher. And that gives us kind of a singular strategy across our spores and other grants that ends up, I think, allowing us to leverage our resources in a more efficient way. We're not using 3 different cloud systems. We're not using 3 different strategies. We have kind of one strategy for data storage. We have one strategy for data acquisition, and we do that across all of our larger grants. And I think that really helps us be both nimble and efficient with our resources. And that really cuts across, I would argue, all areas in the Cancer Center. So hematology, breast, GU, sarcoma, peds, really interact with everyone. And that includes the folks like population health, rad-onc, and some of the areas that cover not just the Cancer Center, but other areas as well. Again, I said tries to help us be nimble. So again, probably should update the timeline here, but when ChatGPT was announced in December of 2022, I would say it was no more than a week before Dr. Johnson came knocking on my door and said, we need to figure out how to publish some work on this and evaluate how useful this is actually in our space. And so at that point, Doug had a medical student, Rachel, that was working with him. And we structured a couple of studies, I would argue, very quickly. And to Rachel's credit, she was able to carry these out, including a prospective study where we took a variety of experts, and we started in cancer, but again, Rachel was very ambitious, and so we pulled in pulmonology and rheum and neurology and some other groups to assess, I would argue, across multiple medicine specialties, the ability of ChatGPT to answer a variety of what we would consider specialty questions in oncology. Both binary questions would have kind of yes, no answers, and things that had a longer description. And so I helped Doug and Rachel structure that project and helped with the manuscript writing. And so again, I just think that having those kinds of data resources are just incredible. And we've continued to kind of move along in this space. But when these opportunities come along, it's nice to have the infrastructure in place so that you can do these things quickly. It was easy for us to gather the data. We already had people that were champions in their areas for these kinds of projects. I won't belabor our shared resource too much, but want to talk about mCODE. Again, mCODE was a project that was developed in 2018, was first released in 2019, lives on top of FHIR, and is a common set of data elements that describe a cancer patient and their journey. And so here you're all familiar that, any 2 EHRs don't usually talk well together. I would argue even in the same vendor system, you might be able to share within that vendor system, but if you want to do anything outside of that vendor system, in this diagram, for instance, use external repositories, data stores, et cetera, to transfer data. That's usually a challenge. And mCODE living on top of that FHIR interface allows us to transfer those data between institutions. And we describe this in the paper that's inset below. But again, it lives on top of this FHIR layer and allows people to build applications that utilize the mCODE, not only to connect to FHIR, but then to know the data elements that are accessed. And this is a diagram that's been shared before, but the current state of FHIR-enabled interoperability for breast cancer. This was a study that was done at MITRE. Lots of data elements when we were creating this were found to be relatively inaccessible in the medical record. And so this was one of the areas that we focused on as we developed mCODE. So mCODE has 6 high-level domains that we divided our data elements within 6 data elements, data areas. So disease, outcome, genomics, treatment, patient, and assessment. And our goal always was not for this to be healthcare institution-centric, but we wanted also to include other stakeholders in the broader healthcare ecosystem, like vendors and payers, research groups, governmental agencies, et cetera. And so this has been just a tremendous progress that we've seen really since 2019. This is the second version. I think we just are balloting now for STU4, standard for trial use 4, but gives a high-level description about the types of data that are available within the mCODE data model. And we were incredibly excited that this was the standard for the new CMS Enhancing Oncology Model to accept digital submissions to CMS. And so it's really taken off. But what I'll say, since I've taken over from Monica Bertinelli as the chair of the mCODE Executive Committee, is that I think one of the reasons for the success of mCODE is how it's interacted with and really been synergistic with the other large projects in the data standards space. So we cover the oncology space. We try hard not to reinvent the wheel and leverage other data standards where they exist, but there are some meaningful gaps in the cancer space. So I would argue, for instance, until mCODE came along, we didn't have really great consensus about how we structured staging. For instance, we had several groups that had their own proprietary staging systems. I'm not going to name any of them here, but how do we, in an open way, think about how we codify staging? Another data element that comes to mind, for instance, is the concept of disease status. So how is a cancer patient responding to their treatment? That's key. It's key for progression-free survival and other outcomes that we all care about. There's not really a place in the medical record to codify that in a structured way. And so mCODE was able to take a stand on some of those things while also supporting up-and-coming projects like the Gravity Project for Social Determinants of Health, and say these are not elements that are specific for cancer, but these are going to cross-cut across different patients and different diseases. So again, I mentioned in 2018, the initial working group was started. 2019 was our first ballot. And if you haven't been through the HL7 balloting process and taken a project through, don't. It's just an incredibly interesting process that I now know more about than I ever thought I would. Our STU1 was released in 2020. In 2021, we released STU2. And then in 2022, we did something called the FHIR maturity model. And with the FHIR maturity model, what we did there was a commitment to vendors to say what parts of our model are most stable. So if you're Epic and you have to decide how much of this do you want to implement, you don't want to spend a bunch of resources implementing something that we're going to go and change in the next month. And so we made that commitment to vendors to let them know, again, in a standard way using the HL7 maturity model through FHIR for each of the data domains, what the maturity levels are. So I mentioned that I think one of the reasons that mCODE has been successful is how it's played with the other large data standard groups more broadly in the community. I think the second has been our ecosystem that we created around our data standard. So again, on the left, we have two groups that really focus on the standard itself, the executive committee, which I lead, in the technical review group, which Sanjay Neha from Yale leads. The implementation side is led by a community effort called Codex. Codex is a member-driven FHIR accelerator. And so this is a group within HL7 where groups can propose new projects that implement the mCODE standard, and then are resourced with project management, et cetera, to help watch those projects go through. And we get feedback from those Codex projects to the technical review group on gaps, especially in the data model, and places where we need to expand the standard. And that closed the loop feedback system, it has just been incredibly helpful to have an organized community where we can get feedback. And then the technical review group makes those recommendations to the executive committee. Executive committee reviews those, and then the technical review group helps implement those changes along with HL7 and the broader standards community. One kind of cut out from Codex is the broader community of practice, which is where most implementation groups start, is joining the community of practice to understand how mCODE's being used in different areas. We have over 55 health systems that are currently implementing mCODE, 7 payers, 10 pharma companies. Lots of EHRs we lump together, other tech companies here. We're involved with lots of professional societies and consortia. I actually just yesterday returned from a joint meeting with NCI and ONC talking about cancer data standards. And again, governmental agencies, ONC included, very supportive of mCODE and the work we're doing there. I'm trying to coordinate with them to to make sure that mCODE lines up with US CDI plus cancer coming out of ONC. And so this has been just, again, an incredible project to be a part of. But what it really comes down to at the end of the day is, what's the utility? Where are people actually using it? And so here are some of the use cases that we're tracking. So mCODE kind of plus data extraction. What this means is that there are data elements in the mCODE data model that aren't necessarily structured always by the electronic health record, either because the technology isn't there or because users choose not to. And I'm not pointing the finger, because at my own institution, our staging rates and the number of times we go through and fill out the very detailed structured staging form for cancer is under 8%, which is abysmal. I would love to have that number higher. It would be great for studies. But mCODE++ allows some methods to try and use NLP and other procedures to go in and structure those data for the user. We've had a couple of projects kind of in that space. EHR endpoints for cancer clinical trials. So we can fill out these electronic case report forms, and we shouldn't have to. And if you live in the informatics space, we shouldn't have to. These are structured data, and we should be able to send those along. And it is just infuriating to me. And so mCODE, we leverage LOINC and the other data standards that underline this. And so EHR endpoints for cancer clinical trials, the iCare data study is doing just this. So we have, I think there are 7 trials using our system to electronically extract the data from the EHR and electronically using pre-populated then electronic case report forms. We don't have to copy and paste the data. I mean, these are just projects that we should just all do as a community. Similarly, integrated trial matching. Trial matching has been a perennial problem in the cancer informatics space. And I would argue that mCODE has gone a long way to defining the left side of the model, the patient side of the data model. A personal project, and would love to get into this in the Q&A portion. I would love to see us take on the challenge of structuring the clinical trial side of the equation on the right side. Would love to see us structure, for instance, inclusion and exclusion criteria and make that a mandate federally to submit to clinicaltrials.gov, for instance. That's a current pet project I'm trying to gain some traction for. Cancer registry reporting, again, we should assist our registrars in extracting everything we can via mCODE from the health record to submit to cancer registries. Radiation therapy, this has been one of the biggest ones, and this really took off. We have coverage for over 90% of patients that receive radiation in the United States have the radiation portion of their healthcare record that are accessible by mCODE because of adoption by the underlying EHR vendors that run radiation oncology practices. They've just completely bought in. They've seen the struggle of dealing with some of the larger EHRs. And so they've adopted mCODE to make an easy way to get those structured, in this case, radiation treatment summaries out of their EHR and available for other electronic health records. Prior authorization, again, near and dear to my heart. I don't like filling out prior authorizations. I don't like doing the appeal process. But on the payer side, they feel the same way. They would like to just get the right data the first time. And so if they can specify what data they need and have that data sent in a structured way to them, and they don't have to dig through notes, that saves them money, is their argument. It saves me time, which is my argument. So this was a synergistic project. And all of these are underway. And so I think I already probably went into this one. This is the EHR endpoint for cancer clinical trials. Again, opportunity to take information directly from, and I was wrong, trial sets, I guess updated the slide here. We're just incredibly excited to support all of these efforts. I should have clicked through these as I was talking, but I get too excited. This is such a passion area for me. Now, I talked about the maturity model, and not all areas of our model are equally mature. So structured genomic data, we have a lot of fields for structured genomic data. Most institutions don't have structured genomic data, so it hasn't been tested. So the genomics domain is least mature. As a summer project, I have incredible interns from Vanderbilt University, almost all in the computer science area, work with me and do this demonstration project to take our structured genomic data. So we get this from a variety of vendors. We just crossed the 25,000 report threshold this week that are structured in our medical records. So think structured like lab values, not PDFs, and expose that via a Fire server, and then do a demonstration project to show that mCODE is able to query those and get those out via mCODE. So this was an exciting demonstration project. I think it was the first in the country to use that part of our I couldn't be prouder of the group, and really want to highlight Vivian Lee in the lower left, who's now at Columbia getting her PhD in informatics. But it was a great summer program, and was great to see that the standard actually worked, and want to give a nod to both Fund for Innovation, Microsoft for help funding that summer project. So we've talked just a little bit about the work of structuring data, making data available to investigators. But a small part of what I do is actually being an investigator as well. So we have a trove of structured data. And so about 5 years ago, we embarked on this partnership between Vanderbilt University and it's been a really fun road to see. The high-level summary of this project, I think, is very straightforward. Again, I'm a medical oncologist, thoracic malignancy, so I focus on lung cancer of the world, which most people know the brand name of because of the commercials. But it's PD-1, PD-L1, and CTLA-4 inhibitors primarily, although we have seen an emergence of lots of biologics that fall under this space because of their modulation of the immune system. But for the purposes of this, we're going to talk primarily about PD-1, PD-L1, and CTLA-4 inhibitors. What's interesting here is the efficacy of these drugs is less than chemotherapy when you look at it from a response rate standpoint. So just on the number of people that respond, fewer people respond to these drugs in my practice than respond to chemotherapy. But what's interesting is they generally are better tolerated, but more importantly, some patients have this incredible response where even after you stop drug, their cancer continues to be controlled for sometimes years. So I have a patient that we stopped treatment on 9 years ago, stage 4 non-small cell lung cancer, and continues to do well with no therapy after 9 years. Now, is that a cure? I don't know. It's awesome that we have to think about that as a question, and we argue about that in national meetings. But what I don't want to have happen, I don't want to miss the opportunity to find that patient because they didn't get a chance to get that drug. And so that was really at the core of this project. How do I figure out who is most likely to respond to these drugs and be one of these outlier responders? And then also, who is most likely to have one of these side effects that are less predictable than they are with chemotherapy? So how did we structure this project? Well, one unique thing, and Peter kind of mentioned this in the opening, unlike other studies in this space, you know, in oncology, we tend to be very disease-centric. And so we did not focus on melanoma or lung cancer or renal cell. We took any patient who's received these drugs. So all comers over a 12-year period, I guess now, we had these drugs in clinical trials pretty early on. And this is the distribution of what our patients look like. So about half of our patients are patients with melanoma. That largely reflects that melanoma had some of the earlier indications, and we had a very strong melanoma program in that clinical trial phase. So we have a lot of melanoma patients. About a quarter of our patients are lung. That's the second and fourth column. We divided them by histology, and about another 15% are renal cell, again divided by histology, with the third and fifth column there. So at that point, I think it looks like a pretty typical mix. And then on the right, you see what the checkpoint inhibitor breakdown is. And what you see is, I think, reflective of most practices. Again, probably with a little bit of a nod to drugs that have been out longer, have a little bit larger representation just because of the longitudinality of our data set. So when we did this study, here's our kind of typical table 1 on the left. I think you can see that we have a male predominance, about 61%. You can see the cancer type listed there, the drug class, smoking, etc. Again, all these seem to be tracking pretty well with clinical trials. We had 2,700 patients in our top-level data set that received checkpoint inhibitors. That meant that we had at least tens of thousands of clinic visits, millions, or if not tens of millions of lab values over the course of their treatment, et cetera. But from an individual patient level is 2,700. And our consort diagram is listed here. And after excluding for specific values for each of the models respectively, we ended up with about 2,000 patients in each of our training sets that we used. We focused on 3 specific toxicities and the outcome of overall survival or response. The 3 toxicities that we focused on were pneumonitis, inflammation of the lung, hepatitis, inflammation of the liver, and colitis, inflammation of the colon, which usually results in diarrhea. We picked these 3 not because they were the most common, because fatigue and rash are probably the most common, but fatigue and rash are usually not reasons to discontinue therapy. I have to consider discontinuing therapy for patients with colitis, hepatitis, and pneumonitis. These are also not necessarily the most severe. Again, Dr. Johnson, who I showed earlier, published in the New England Journal, our experience with checkpoint inhibitors and cardiac toxicity, for instance, including very severe myositis, which not infrequently is fatal. But we chose to not focus on those endpoints, even though they're very severe, because they're very infrequent. So this was the nice mix between giving us some sort of event frequency and the clinical outcome of oftentimes being clinically important because it would change therapy. So how did we engineer this? Well, we had all of our data in our electronic data warehouse. It was already OMOP mapped, and so we pulled those out. We aligned to data standards. We did have some fields that weren't structured in the medical record, so we had a team of abstractors for things like smoking history and some other fields, features that we were interested in. And then we brought the 2 groups back together. The piece that's not here is we have a separate kind of work stream within the project that's also looking at using imaging to predict response and toxicity. Dave Smith is kind of leading that group with me, and I think his group will be publishing that paper kind of shortly. But the end point will be to bring the 2 models together and see whether imaging adds incremental benefit to the EHR data, which was the primary focus. So what was our methodology? So one of the things I really appreciate about this project is our 2 groups were able to do more together than either of us could have done alone. So on the academic side, we were able to be very academic about, I think, how we structured this data science project. Most of my role in the early years were focusing on making sure we weren't leaking data into the model. And on their side, they wanted to make sure this was reproducible, so they made sure all of the data were clean and that this pipeline was documented in a way that you would expect from a commercial entity. So model inputs are taken in before the first checkpoint inhibitor administration. That's important, again, because we've seen several projects that are looking at predicting after ICI administration, and they include lab values, for instance, potentially just before the radiology, and to me that gets really messy as far as predictions go. So we thought the cutoff is right before infusion time, and we have a variety of windows that go back 60, 100 days or a year into the past. And again, these are all structured data that I would argue we're collecting at all of our oncology practices, things like ICD-10 codes, medication history, radiation history we included. It wasn't a feature that actually came out, so that one's easy. And then we used a typical cross-validation strategy, but we did for each of the cross-validations do an inner loop. Again, just this wasn't the largest data set, so we wanted to be extra sure that we didn't get bias just from a bad sampling. So there's an inner loop for each one of those 5-fold round validations. So here's the results. We were excited to share these both in the publication and at IO360 this year. What I usually tell my students in informatics is if we're talking about translational AI research or translational ML research, especially in the healthcare space, the AUC is not the full story, because rarely in healthcare are you weighting sensitivity and positive predictive value equally. And I feel like this is the soapbox that, again, I always stand on in my project. So I'm not going to read all the results. You can see them there. But I do want to point out that, again, as a PI and someone that dabbles in data science but sees patients, what I want to see in these charts of feature importance on the right is I want to see a couple of features that make sense clinically to me. So let's pick the bar that stands above the others, which is colitis. So what is the biggest factor in our model influencing colitis? It's co-administration of anti-CTLA-4 inhibitors, or anti-CTLA-4 directed therapy, double negative. Does that make sense? This is not only does it make sense, this is evidence-based. We know this is true. It's a great result from my standpoint. This is a positive control. Similar with pneumonitis. Okay, lung codes is a conglomerate endpoint of codes like COPD, emphysema, et cetera, interstitial lung disease, and it is the most weighted feature in predicting pneumonitis. That's not enough, though. It's all of the other features that I don't necessarily, as a clinician, know how to integrate together to make the final prediction. So I couldn't ask more for how our features looked. One, because I had those good positive controls, and two, because what we came out with was largely things like BMI, albumin, alkaline phosphatase, things that we're already collecting in clinic. So again, to my executive groups, it's always nice to just show the clinical utility and really simple to understand stories. So if I had 200 patients and I needed to prioritize 100 of them for a hypothetical study, we could use the 1-year overall survival model and move from a random selection of our patients that would have a 52% overall survival at 1 year to, if we pick the top 100 informed by the model, that would give us 68% overall survival. Now, this assumes that you're okay with losing half of the cohort, but maybe that's what you want in a phase 1 clinical trial if you're a pharma company. Maybe that's actually okay. Similarly, for pneumonitis, background rate is 9%. If you took the top half, the half that had the least chance of pneumonitis, you could actually run a study then that had a 4% pneumonitis risk based on the model, which again might mean that you move forward with an asset that you wouldn't otherwise move forward with if you were a pharma company because of the toxicity profile, and that's one of our endpoints. So this was a random forest model, again, showing the overall survival model, overall survival differences with different time horizons. And one of my colleagues likes to joke that this isn't surprising. This is the same thing that we see in the weather. So at 100 days, we're better able to predict than at 1 year, than at 3 years, than at 5 years, but we give it a shot. So I always like that analogy. And as it shows here, this is a random forest model. We did what I think every informatician does, is we tried every model, right, and picked the least flexible model that held our results. And for us, that was random forest. And these are the results for overall survival. Again, AUC here may or may not tell the whole story. We were pretty pleased with the outcomes of this, though. But for me, the real story is around clinical utility. So it means that if I see that someone has a high risk of toxicity, they have a low risk of overall survival, I might have a lower threshold to change their treatment or stop treatment, especially if they're having a toxicity. Similar numbers here for pneumonitis, hepatitis, and colitis. Now what we've done is we've separated them by tumor type. And I will warn you, everyone has asked for this, but the numbers get really small here. So the confidence intervals are huge. There was a reason we did this as a pan-cancer study, where we would welcome the opportunity to collaborate and combine our data with others to try and investigate some of these things to see if the pneumonitis risk in lung cancer, for instance, has some nuances. But everyone asks always for the breakout there. We've also been working on time on treatment. This is kind of our surrogate for progression-free survival in real-world data. Again, this is a challenge, but everyone wants to know how long you were getting benefit from that drug before you had to start something else. So again, I would say this is pretty early, not yet ready for prime time. The AUCs are certainly still several points behind where our other studies are. This was exciting. And so one of my colleagues at GE Healthcare started this before telling me about it, and I couldn't be happier because I would have been nervous going into it. But the University of Essen in Germany reached out, and they have a similarly structured cohort, and were in contact and said that they would be interested in running our models on their data set. And so we ran the overall survival and hepatitis models. This was an international external validation, which is a high bar. And like I said, I was really nervous that this would have gone really poorly. But we retained 90% of the performance on overall survival, and we retained 86% of the performance in hepatitis prediction. That went way better than we thought it would. Again, both data sets, these were not clinical trial patients. These were just routinely cared for patients. And so I think really circles back to that assertion that I made around these are data that are collected already. This, from a rollout standpoint, shouldn't be difficult because we're already collecting these data. So I won't spend too long on the results summary because I think we've talked about it. Going forward, though, on this project, what we're excited for is the next phase. I would love to do a silent prospective study looking at these data going forward in a different setting. One of the risks is always, has the field changed and does current therapy change the impact of this model? I would love to validate that prospectively. It could be in a silent way as part of a clinical trial. And then I think our two main stakeholders and end users here are probably clinical practice, certainly number one, but I'm also excited about the opportunity to partner with pharma and potentially salvage some drugs that might work for some of our patients that we could find and identify in a precision approach to match patients with drugs using models like this. So for instance, if an asset has a very high toxicity profile, but we can find the patients where they're least likely to have a toxicity and they would gain benefit, I would love to have another drug for those patients. So with that, I just want to thank lots of folks. Our data science shared resource team, this is kind of like my little SWAT team. They're absolutely fantastic and just help us get the work done. mCODE, the Executive Committee and Technical Review Group, and a special nod to Monica. And then with that, happy to answer any questions. And thanks again for the opportunity. It's really fun.
Wonderful. Well, I want to give the students a chance to ask questions, but I could start us off while people think of some questions. So first of all, really great talk. I thought it was really interesting from many perspectives. And I do have actually several questions, but I'll just start with one for now as people come up. And that's in terms of the AI ML modeling. I thought it was really interesting what you said, that AUC doesn't tell the whole story. And that's definitely the case because it's essentially evaluating all the decision thresholds. But when you're translating to the clinic, you're choosing a particular decision threshold. So I was wondering, you said all the metrics are not weighted equally. Like for clinical practice, you might want to optimize sensitivity over PPV, or you make these decisions. So I'm just wondering, in terms of you have all these possible decision thresholds. Do you optimize the decision threshold to, say, optimize sensitivity?
Yeah, it really is a great question, Peter. And this is one of my favorite hints to get my students to ask questions in grand rounds is around clinical utility and decision thresholds, because I feel like it's the thing that people leave out of their data science presentations. So when I think about decision thresholds here, I think about it in terms of clinical utility and actually changing practice. So for instance, let's say I'm predicting hepatitis, which we are. What can I do as an intervention in someone that I predict is going to have hepatitis? Well, normally I would check their liver enzymes every 3 weeks on treatment. Perhaps though, if I set a threshold and the model triggers, maybe now I'm going to check your liver enzymes every week so that I can do an earlier intervention. So then I have to weigh again, if I'm titrating for sensitivity, I have to weigh what's the cost of those 2 extra CMPs to the system versus what's the savings of those 2 extra CMPs saving an inpatient admission, for instance. And I would argue that the CMP is not expensive in the scheme of things. And so in that case, we're happy to titrate to sensitivity there. And if you can give me a positive predictive value of 50% or 40%, that's a pretty good value proposition I can take to our cancer center and say, I would like funding to pay for the CMPs for this select population because I think I can keep them on trial longer. And that's a great outcome. Now, if we do the same thing with pneumonitis, now maybe it's CT scan. CT scan's more expensive. And so I'm going to have a different threshold for that. So I think that it's not even that I'm picking sensitivity over positive predictive value. I think you have to look at each of the models, and then you have to look at the setting in which they're applied. And I think the titration piece, the tweaking of the model and the balancing between the sensitivity and positive predictive value probably is actually an implementation question at the last phase where you're actually doing this in the healthcare setting or in the pharma setting.
Wonderful, thanks. Other questions?
I'll go ahead. Thank you very much, Dr. Osterman. I really enjoyed your talk, and all 3 topics were very interesting. I'm the chief science officer at our cancer center here, and we're a small cancer center, kind of on the frontier in the Pacific. And our cancer center doesn't have a medical center tied to it, an academic medical center. So we do all of our clinical trial work and community partners. And we've been looking at tools that are where you use AI to mine, the healthcare systems to find patients for clinical trials. And it's if you're not the owner of the medical system and you want to interface with like 8 systems and use tools like maybe you're familiar with like Deep6 and those type of solutions, incredibly expensive for a small cancer center to take on. And it sounds like mCODE is potentially a tool that would allow you to do your own mining across everybody. And can you contrast a little bit of how mCODE would help in that situation versus going with these external vendors?
Yeah, I will try not to be like the pessimist here, but I will say that the technology actually is probably the easy part, right? It's usually the contracting and the legal and privacy pieces that end up causing the problem. So I think that mCODE offers a way, if those other pieces are in place, to define the patient and share that across institutions, certainly. Jim Chen, who's at Ohio State now works at Tempus, has shown through a project called GenomeX the ability to do that, to take the mCODE genomics module and show that a subset of that for the genomics data can transport genomic tests again. So some of the harder to transfer data from one center to another. So I certainly think so. But the other, I guess, kind of open source standard, or maybe not open, maybe open source isn't the word, but standard that's emerging that we'll see coming up quickly is TEFCA from ONC. That ideally, I think the vision of that is this nationwide interoperable health information exchange, HIE, that will allow your cancer center to make a request in a standard way to another center and get those data back in a standard way. And I think that at the base level will be US CDI elements. But I would hope that ONC also leaves the door open for US CDI plus cancer, which is largely informed by mCODE and other data standards. So I think TEFCA is probably the best answer there.
Great, thanks.
We have a question in the chat, which I can read aloud. So do you have access to or collaboration to real world data and integrate this into your model to predict response?
Yeah, it's a great question. So one of my fellows, Joseph Vento, is graduating this summer and finishing his master's and finishing his fellowship, his master's in biomedical informatics. And he's spent his master's years focused on using the cancer-linked data. So I think it's the largest set of real-world cancer patient data in the United States. Flatiron might be bigger. I think CancerLink's bigger. I might be wrong. But one of the two are certainly the biggest, I think, probably real-world data set for cancer. And it's been incredible, some of the discoveries that Joseph's been able to do. The challenge in this scenario for this project is really price. So for the longest time, CancerLink didn't have an option for any commercial venture. And because half of our project is backed by a commercial venture, that was off the table completely. We're not sure whether or not the data from Flatiron would be able to make an impactful difference in our model. And at this point, now that we have results, I think we have more interesting groups that are interested potentially in sharing data, or like the Essen example, interested in us providing the models and them validating our models on their data, that the value proposition might not be there. But I think, you know, Flatiron, CancerLink, other data sets like this offer tremendous opportunities for discovery. So it wasn't because we didn't think the data were valuable. The value proposition was a little more challenging since, again, part of our partnership was a for-profit entity.
Other questions? I guess I'll ask one last question, which is, so if you have an oncology clinic that wants to adopt mCODE, what are the differences and challenges versus a clinic that already uses FHIR versus one that, you know, might have their data Epic but doesn't use FHIR?
Yeah, it's a great question, Peter. So one of the comments I made at the ONC NCI meeting earlier this week in DC was, you know, the ONC has a mandate for EHRs to implement FHIR, and they can stand up front and say that 90%, and I'm probably getting the number wrong, but it's a high number. It's greater than 75%. I think it's 90% of the EHR vendors, you know, officially have implemented FHIR and can send the US CDI data elements over FHIR. And that sounds great. And I think as a natural effect of that is that vendors think that FHIR is available in all these healthcare systems. And that's not what we find, certainly. And I don't know how it is at your collection of systems, but even at Vanderbilt, we don't stand up a FHIR profile unless there's a use case where it's being required. No one goes through the work of just making the connection for the connection to sit there. And so one of the things that we proposed is we really need to measure not just the vendor implementation, but the institution implementation of FHIR, and then the number of projects actually utilizing those resources. And I think we need all three levels of that pyramid in order to understand the FHIR adoption. So your question to, you know, an organization that has implemented FHIR and hasn't, I would argue even the organization that has implemented FHIR might still have some FHIR work to do to expose all the profiles. And again, I showed the project with my students over the summer. I don't think anyone had really implemented the FHIR genomics standard on top of Epic, for instance. So that was non-trivial to get that part of it up, even as a demonstration, because that part's just new. That's been a relatively emerging standard for structured genomic data, very different than you know, patient data, like the patient demographics and data model. So certainly more challenges on the FHIR side. Once you get the FHIR model up, the rest of the mCODE pieces just come together. That part's quite a bit more straightforward. But if groups are interested, I would always just point them to the community of practice. We host a monthly webinar. It's a great place. Every week or every month, sorry, we highlight one of the implementers and give them a chance to kind of share what they've done, both the challenges and the successes for the community. And that's always where I point people to start.
Okay, thanks so much. Well, if we don't have any other questions, then I think let's give Travis another round of applause. And are you okay with us posting this online?
Of course.
Okay. Well, we'll be back in touch for the beer stein. Thanks so much for your time and have a great weekend.
Thank you again for the
Great. Thank you,
invitation. That was
Travis.
great. Take care.
Okay, bye-bye.